Hi, I'm Angelo Mendoza of DVM3A. Today on Microbiology 151, we will discuss the pouring and inoculating culture plates. Let's start. The first step is to collect samples from the animal secretion, either feces, saliva using cotton applicators, and urine using tub containers.
When getting a urine sample, get a clean tub and wait for it to urinate. Make sure to catch it on time.
Transfer it in a vial and make sure to close it tightly. The next step is heating the Erlenmeyer flask containing the agar.
Make sure that the water is in 65 degrees Celsius. Cool the flask to about 50 degrees Celsius.
Next step is pouring of the medium to the petri dish.
Do not forget to flame the mouth of the flask between each pouring.
Flaming should be sufficient to destroy the microorganisms.
Place sterile petri dish on the laboratory table by sliding them on.
Do not put the petri dish directly on a table because if the cover is lifted even slightly, the air current that carry dust will enter and may cause contamination.
Raise the cover of the sterile petri dish just enough to get the mouth of the flask in, and pour the sterile agar into the petri dish. It will be filled with 20 to 25 ml. Gently rotate the dish so that the agar covers the entire surface of the bottom.
Lastly, place the dish in an undisturbed place to harden the medium.
The agar will harden in about 15 to 25 minutes at room temperature.
That is how we obtain samples and pouring agar plates. Next, let's proceed on inoculating culture plates. First is to flame the entire wire portion of the inoculating loop over the alcohol lamp until it turns red. Cool the loop until the reddish glow of the metal vanishes.
Dip the tip of the loop into the specimen, which is the urine. Flame the mouth of the agar plate and raise it, just enough to permit the entrance of the inoculating loop, and streak the plate.
Do not dig up the agar. The surface of the agar should be smooth when you are finished. After inoculating, label the bottom part of the petri dish with initials and date.
And lastly, incubate all the plates in an inverted position in an incubator.
And that's it. I hope you are guided with proper pouring and inoculating culture plates. Again, this is Angelo Mendoza of DVM3A for Microbiology 151. Bye!